Our focus is in cell engineering for a new generation of cell-based therapies and cell-based medicines. We're a public company on the London Stock Exchange, so hence the forward-looking statements. This is a hybrid model. I'm going to talk about the life sciences business, which has been growing at about 24-25% per year, very high margins. I'm also going to talk about our proprietary program called CARMA, which is a rapid manufacturing solid tumor product and platform utilizing CAR therapies. We're based in Gaithersburg, Maryland. We have about 55 employees, most of whom have advanced degrees. So the basis for this business is based on a technology that MaxCyte invented and developed. It's called flow electroporation. What it does is it can engineer large volumes of primary cells. We're talking about 1 × 10⁻⁻⁻ cells in about 20 minutes. We have another system that can get 2 × 10⁻11 cells in about 30 minutes, which is used for viral vector manufacturing and protein manufacturing. We have focused all of our attention on mammalian cells and principally on primary human cells. We can load basically any molecule into these cells, DNA, RNA, protein. You have it, and the business model we have is to license this technology to companies who are doing cell therapy. It's more than a technology. We also have a worldwide group of scientists that work with our partners, identify the endpoint they're trying to get to, whether that be a biological endpoint or a clinical endpoint, and we develop our cell engineering technology to fit their needs. Decades of experience. We invented this technology. The group that's still within MaxCyte is the core of what we do. And we also have significant regulatory experience where we're also able to support the clinical development with our partners pretty much anywhere in the globe. So we partner this broadly. Everything is non-exclusive. We have over 80 licenses to our technology that have been granted. 45 of those licenses, over 45, are licensed for clinical purposes. And we now have announced recently 5 commercial licenses. These 5 commercial licenses total about $450 million of milestones, pre-commercial milestones. And our partners include Kite, they include Precision Biosciences, and they include Casebia and CRISPR, among others. You can see some of the other partners that we have that are either research or clinical programs that we are developing with them. So let me focus now on CARMA Cellular Therapies. We recently made a decision to find independent financing for this group. This was the reason we went public in the London Stock Exchange. And we think we have developed a program and a technology that is going to solve a number of the problems in this field for the next several decades. We began this program about 5 years ago, and it's an autologous messenger RNA-based CAR therapy. It's streamlined manufacturing. We manufacture this product in less than 1 day. Talk about that. It's we think, a very novel approach. We're really excited about this approach in that we're agnostic in terms of the target or the molecule. We can load RNA to express a protein. We can have that protein be a TCR or a CAR. It could be a siRNA. It really doesn't matter. We work with partners to develop therapies that make sense based on the ever-emerging work that's being done. And Oz just talked about several things going on at Penn. We see this field moving very rapidly. There's 2 big issues here. One is these therapies are becoming much more complex, but at the same time, you've got an issue with patient access. You've got to make sure that it can be manufactured, and they can be manufactured at a cost that's reasonable, and it can be absorbed by the medical system. So there's these two competing forces that we think MaxCyte's right in the center of, and we think CARMA is one of the answers to this issue. Our lead candidate is MCY-M11. It's a mesothelin-directed CAR using messenger RNA. This platform is focused primarily on solid tumors, and we'll talk about why we think that's going to be an interesting approach given what we are able to do with this technology. So this comes from quite a number of years of working with some of the leaders in the field, certainly with Penn and National University of Singapore, so both Carl June and Dario Campana. Our preclinical work in CARMA was done at Johns Hopkins Medicine in Baltimore, right up the street from our location in Gaithersburg, Maryland. We have 2 clinical sites going on right now. One is the National Cancer Institute, and the other is Wash University in St. Louis. This program is off the backs of over 20 patients being dosed with a mesothelin-directed CAR by these institutions. So this is one of the major features, if you will, of our technology. What we do is we apheresis patients. We do a very simple phycol step to polish the PBMCs. We then transfect the PBMCs with messenger RNA. We then aliquot and cryopreserve. A single apheresis will produce, depending on the doses, anywhere from 9 to 20 doses, which we think is going to be important for treating solid tumors. We don't believe that solid tumors are going to be, and I think that Oz actually talked a little bit about this, where it's kind of a single shot. You're going to have to do it with combinations. You're going to have to do it with repeat dosing. And we think mRNA is the main answer to that. So this process takes less than 1 day to make those multiple doses and give them back to the patients. The benefit of that, obviously, is being able to have it closer to the patient in a transfusion medical center, if appropriate. It reduces the cost. It reduces the amount of time it takes to produce the cells, and therefore the cost for husbanding those cells through. And most importantly, what we're hearing from clinicians is we want to get the time to treat down. Right now, that can be anywhere from 4 to 6 weeks for a patient. And as you know, if you're an oncology patient or have one in your family, that's a very, very long time to manage that patient. And therefore, we believe that there's going to be a huge need to really figure out ways of treating these patients much, much faster. If you're not familiar with messenger RNA, it's significantly different than virus transduction. With virus transduction, you're literally delivering the DNA into the nucleus with a virus. DNA makes RNA, which makes proteins, of course. What we're doing is we're loading the messenger RNA into the cytosol and expressing the protein. That protein is active for somewhere between 5 to 7 days, although we have seen it in circulation for upwards of 2 weeks. We believe that that's going to be important to go after targets that may be typically concerning with on-target off-tumor toxicity, and what we believe we can do with repeat dosing and the prospective, if you will, management of toxicity to redose these patients and stay within that therapeutic window. So again, we can deliver immune cells against any relevant target. Again, we're agnostic in terms of the target. It's certainly amenable to a number of different combination therapies, including loading molecules into the cell that could either be checkpoint inhibitors or additional proteins or additional CARs. So we have worked with up to 5 different mRNAs at one time in certain applications. So we know this is conceptually doable. And the question is, what is the combination that will be most effective for treating these patients? And again, with non-viral RNA as opposed to virus, we believe we'll be able to multi-dose these patients over a longer period of time, for instance, once a week over the period of 3 to 4 months. This is our current pipeline. We're in clinical trials right now with an intraperitoneal infusion, treating peritoneal mesothelioma and ovarian cancer. Right behind that is a IV version of that. It would be a basket study going after colorectal cancer, non-small cell lung cancer, a number, and I'll talk about that in a second. And we have a number of other targets that we're investigating right now that we'll be talking about in the future. The lead candidate is MCY-M11. The target is mesothelin. Many of you are familiar with this target. What's interesting about this target is how broadly distributed it is across a number of very important cancers, which we think provide us an opportunity to treat. We also believe that the toxicity could be relatively low, given the fact that it's expressed on what we believe to be non-critical tissue, even though we have an mRNA approach, which, unlike a virus approach, which would keep that cell on surveillance all the time, this has a relatively short-term half-life to treat these patients. And you can see by the IP and IV the focus of the company, and certainly in a basket study, we believe there's going to be some really interesting work that we can be done. This is a target that a few years ago people said was not an important target. We did focus in on it a number of years ago, and now you're seeing quite a bit of work that's being done in the field, mostly in combination therapies. But this first program we have is wholly owned by MaxCyte. This is one of the few cell therapies that actually have come from a company, not in association with a university. And so MaxCyte owns 100% of this asset. And again, we're looking for partners to help us develop it on a clinical basis. Some of the preclinical data, one in particular was some work that was done at Johns Hopkins. The green line is a 6 dosing of the karma. This is in immune mouse. So this is just the CAR itself, the PBMCs transfected with CAR. There's no contribution in this study by the immune system of the mice, and you see a distinct survival advantage over 6 doses versus 3. So again, we're very interested in being able to multi-dose our patients to see if we can have an important progress against this horrible disease. The current clinical trial is at standard by 3x3. We're working through the second dose level cohort. It's currently enrolling. We have seen no dose-limiting toxicities or any safety concerns. We'll be announcing moving into dose level 3 sometime before the end of the year, and we've talked to investors about having a full readout of this program by the middle of 2020. And again, this is interperitoneal dosing. When we think about this in the future, and this is a platform, we think about things like preconditioning. We also think about combination therapies with checkpoint inhibitors. As I mentioned, we also are thinking about developing different molecules that can be loaded and expressed on these cells at one time. So there's a number of different ways we want to take this further into the clinic. We've been able to demonstrate the utility of the manufacturing. Now we're looking for partners to help us work through determining the clinical utility of the molecule itself. So from an investment perspective, we have an experienced leadership team. It's a very, very broad platform. The Karma platform has not been licensed. It's still owned 100% by MaxCyte. And again, we're going to have some near-term MCY-M11 clinical readouts. This is the team, some of whom are in the room. Kathy Wexelman is here, Michael Kuo. I don't see anyone else from the team. Very experienced team coming from the National Cancer Institute, both our head of clinical and CMO, as well as Takeda, Infinity, and most recently they both have joined MaxCyte. So a great leadership team that's focused on moving this into the clinic and beyond. So again, we believe this is a disruptive technology because of the way we can manufacture this product. We think that this will eventually go into close to the patient treatment, if not in transfusion medical centers, who have all those same unit operations. The only thing they don't have is the MaxCyte electroporation technology. Again, lowering cost of goods sold and really changing the way we think about manufacturing and delivering cellular therapies. Again, it's a very flexible platform in that we can use it with combinations, and we can also use it with virtually any target for CAR therapies and for TCRs. And again, the cell product itself is unique. It's PBMCs, mRNA, non-viral, and again, multiple dosing from one patient apheresis. So readouts for this one program we just announced dosing of the second cohort. Additional cohorts dosing in phase 1 will be sometime in the fourth quarter. We'll be announcing clinical results. We just announced the trials in progress at ESMO. We'll be at the SITC meeting and other meetings as data matures. And then again, top-line results from this phase 1 trial sometime in the middle of 2020. So thank you for your attention.